I'm Linn, Principal Investigator of the Diabetes Research Group at Justus Liebig University, Gießen, Germany. Pancreatic islet transplantation is a treatment for type 1 diabetes mellitus. Experimental islet transplantation is performed to the kidney capsule, while clinical programs prefer the intraportal route. Therefore, we believe that more research on intraportal islet transplantation is needed in preclinical models. For example, islets are exposed to blood as the transplantation site is intravascular. Moreover, the microsurgery technique needs some experience. For example, after the injection of the portal vein, bleeding can occur, or there may be issues with anesthesia or with the maintenance of the body weight of the recipient mouse. Therefore, we think that visual explanation of the procedure is crucial. This video focuses on the intraportal route of islet transplantation. To achieve normoglycemia in diabetic mice, it can be used for syngeneic and allogenic transplantation in black mice, but also in xenogenic models in nude mice recipients.
Remove the isolated islets from the incubator and place them under a flow bench. Center the islets with the circular swinging motion of the Petri dish. Fill 100 microliters